Okay, so let's get started. Today, excuse me. Today we're going to start a topic that's a little bit.
I had the same excuse she has.
All right. So today we're going to start a bit of a standalone two lectures. This one and the next one are related to metabolism, drugs, xenobiotics. It's one of my favorite lectures to give. It's really where I get to start talking to you guys about toxicology. And again, this we're going to note, we're not using if you're reading the textbook, we're not using the typical biochemistry textbook. We're using something on Access Pharmacy called Cassart and Duell's Toxicology. There's some sections on cytochrome P450s and the conjugation reactions that are used in phase 2 metabolism that we're going to talk about today. So today, we're going to define what a xenobiotic is, be able to describe the purpose of the 3 phases of xenobiotic metabolism, talk about the reaction of cytochrome P450s, and describe the characteristics of the actual enzymes, describe the various reactions that occur during phase 2 xenobiotic metabolism, and then describe the overall effect of phase 1 and phase 2 xenobiotic metabolism. So first of all, what is a xenobiotic? A xenobiotic is anything that is foreign to the body, any compound. It can come from a variety of sources. It could be a drug, for example. These are all xenobiotics. It's something our body does not produce, and so we have to deal with it because certain xenobiotics can be very harmful to our bodies. So there are many different types. There are natural xenobiotics. These are, if they harm us, they're called toxins. They are compounds found in nature. They often come into our body either through our diet or we're exposed to them in our environment. Then they can be also man-made. And the man-made ones, typically we talk about toxicants, which are harmful compounds to our body that do not occur in nature. These are things like pollutants, pesticides, anything derived from oil, petroleum, plastics, and then other compounds that aren't toxicants, typically, food additives and drugs. So there are many different kinds of xenobiotics, and we don't have time today to talk about every single thing. So we're only going to talk about how we actually deal with them once they come into our body. We'll just give an example of a natural toxin. This is a compound called solanine. It causes GI and neurotoxicity that can lead to coma and death. It's produced by members of a plant family called Solanaceae. The most characteristic one is the belladonna plant, which is the flower and plant is given right here. It's an interesting plant in that during the 14th, 15th century, this was actually extracts were used to dilate women's pupils as a cosmetic because it was thought that these dilated pupils were making women more seductive and were a great type of cosmetic. However, these women, if they used it too much, went blind. And so it wasn't quite the best idea, and the use of this compound took a while to wane, but eventually it did. And so we don't use that anymore. But these solanines we're actually exposed to very frequently in our everyday life outside of anyone who likes to play with the belladonna plant. So we actually see these solanines when we eat things like potatoes, eggplant, tomatillos, tomatoes. These are all in the same family of this plant, this belladonna plant. However, the levels of the edible parts are very low, and so our bodies can easily metabolize them and get rid of them. And so that's the whole idea of the xenobiotic metabolism, is try to deal with these compounds so that they don't harm our bodies. With the exception that with these same plants, potatoes, tomatoes, eggplant, their exceptionally high levels are found in green fleshed potatoes, so that's why you aren't supposed to eat those, and also in the non-edible parts, the leaves, the flowers. There's reasons why we don't chew on a tomato plant, and this is one of them. So when we talk about xenobiotic metabolism, we're looking at ways in which we can convert lipophilic compounds into things that are easily excreted. We're trying to make them more hydrophilic and get them out of our body. The detoxification and inactivation will convert these chemicals typically into a less toxic form. So the major sites of xenobiotic metabolism is the liver, which is the primary site, and it's very rich in the enzymes, especially the cytochrome P450s that we'll talk about in the next section. And then there are other sites of metabolism which are also very important, including the intestinal wall, especially for sulfate conjugation. They also contain esterases and lipases, which are important in prodrug metabolism, as well as the lungs, which is the first site of metabolism for anything that's inhaled by our body. It's also very rich in these enzymes. There is some metabolism that also occurs in the kidney, the placenta, skin, and adrenal organs. Typically, when we talk about xenobiotic metabolism, we talk in 3 phases. The first phase alters the activity of the compound to make it more polar. The second phase, you alter the xenobiotic to make it more soluble, and you usually mask certain functional groups, which are usually harmful. And then the third phase is the actual transport of the xenobiotic out of the cells, either into the feces or into urine. Now today we're only going to be concerned with phase 1 and phase 2. Phase 3 we're going to go over on Friday. So let's look at the first 2 phases. Phase 1 typically involves a hydroxylation reaction, whereas phase 2 involves the conjugation reactions, where a xenobiotic is attached to various polar compounds. Overall, the idea is we're going to make the compound more water soluble and let it go through excretion. However, when we do this, we're also trying to make the compound less bioactive. We're trying to make it so that it does not harm our bodies any further. There are instances in which compounds actually become activated by the whole process of xenobiotic metabolism, and this is called bioactivation. So in most cases, metabolism will lessen the toxicity of xenobiotics. However, some xenobiotics are activated to a more toxic state by the process of metabolism. So these inactive xenobiotics will then become active, and this inactive xenobiotic is often called a protoxin. So this bioactivation usually occurs after phase 1. So we also can take advantage of this principle. Sometimes when we design drugs in a prodrug form, so it actually has to be metabolized before it is actually active. And so we use these metabolism enzymes to help in our drug design. So an example of bioactivation is a compound called aflatoxin. These are naturally occurring mycotoxins that are produced by many species of Aspergillus fungus. They're found usually on moldy peanuts, rice, corn, and other crops. So this is basically what it looks like. When you see this blackish green corn, most people wouldn't eat this. However, if you're doing large processing of corn, you don't have time to check every single kernel. They're going through this large conveyor belt inside of a factory. Easily, if there's this moldy corn, it can get into the process and into the process. In some areas of the world, aflatoxin toxicity is very common. However, in addition to it being this byproduct of this mold, is also a very potent liver carcinogen. So having too much exposure to aflatoxin B1, for example, primes the liver for cancer. Inside of the liver, aflatoxin is turned over into a more toxic compound through phase 1 metabolisms to an ultimate carcinogen, which can directly attack an adduct onto DNA, damaging the DNA and cause mutations leading to cancer. However, there are other phase 1 enzymes and phase 2 enzymes which can help us deactivate this ultimate carcinogen. Now, when there are problems with these other enzymes, the ultimate carcinogen will build up and you'll get more priming for cancer. So there are ways in which our body can deal with this. However, in excess, our body becomes overcome by it. So let's start looking at phase 1 metabolism. This is often called first pass metabolism. Typically when toxins come in, they're either inhaled or they're eaten or they go through our digestive tract. It gets into our blood and then gets filtered in the liver. And then inside the liver, the phase 1 metabolism occurs. So that's really why it's called phase 1 or first pass metabolism. Enzyme type reactions are typically hydroxylation reactions. The purpose being add a polar functional group to increase the molecule's polarity. We will also provide a site for phase 2 metabolism to occur as well. Now, the hydroxylation reaction is a review. We've seen this when we were looking at cholesterol synthesis. Basically, you have whatever your substrate molecule is, here abbreviated by RH. You have oxygen, NADPH, and then you add one of those oxygens as a hydroxyl group onto your substrate, and you produce water. So this is basically an addition of a single oxygen atom in the form of hydroxide. And the hydroxylation will make the compounds more hydrophilic. This is performed by cytochrome P450 dependent monooxygenase, or just abbreviated as cytochrome P450s, or sometimes just as CYP. So cytochrome P450s, or CYPs, are enzymes that contain a heme group as a functional group. During the reaction, the P450s go from a reduced to an oxidized form, so it's a redox type reaction. The highest levels of these CYPs are found in the liver, but they are also found in other tissues. They're located on the smooth endoplasmic reticulum or in the mitochondria, and they are membrane bound. The cytochrome P450s have a very broad substrate specificity. They'll recognize aromatic compounds, benzyl compounds, aliphatic, double bonds. There's many different types, which is very good because in our daily life we're exposed to various types of compounds from our environment, whether it comes in our diet or through a drug or whatever. And so having the capability to handle multiple types of compounds, multiple organic structures, allows these enzymes to start the metabolism. So cytochrome P450s can be directly induced. They're usually induced by the presence of their substrate, so it increases the transcription, and you have an increase in the number of the actual enzymes. They're also induced by drug metabolism. So approximately 50% of drugs that humans ingest are metabolized by cytochrome P450s, and then the levels will go up. These enzymes will also act on carcinogens and pollutants, and they can biotransform substrates, however, into toxic compounds. So the cytochrome P450s, there are multiple isozymes, meaning there's multiple forms of the enzyme. So it's really, when we talk about these, as a super gene family. There's about 8,000 of these P450 genes in our that are known, which is then divided into 368 different gene families and 814 subfamilies. Humans, it's us, we contain 18 of these, 43 subfamilies, and in our bodies we contain about 57 of these known genes. When we look at cytochrome P450s, we usually say CYP as the abbreviation, then the family is given as the 1, the subfamily is given a letter, and then the individual member of that subfamily, so the individual identity of that enzyme, is then given another number. So CYP1A1 means that this cytochrome P450, or CYP1A2 in this example, is the second individual member of the A subfamily of family 1. So when we look at all these different types of CYP enzymes, around 50% of drugs that humans ingest are metabolized by some isoform of cytochrome P450. The majority being in the family of 3A4 or 3A5. Another one that's very common is CYP2D6 and CYP2C9. Then you can see all of these other ones that are acted upon have just a very small fraction of the drugs that we take, and then some, there's almost about a fifth of drugs that aren't acted upon by these cytochrome P450s, but by other phase 1 drugs, phase 1 enzymes. So each family and subfamily will have specific substrates. CYP1, CYP2, and CYP3 are especially important for drug metabolism, with CYP3A being the most important in drug metabolism due to its high levels in the liver and the intestine. So looking at some examples of substrates, inhibitors, and inducers of CYPs. So each of these circles represents one of the CYP families members. So we have 3A4 here being the largest, and the little percentage inside tells you roughly about how many percents of drugs are acted upon by CYP, for example, 3A4, so about 30% of drugs. Up here are some of our more common drugs that are metabolized by it. So for example, 3A4, cyclosporins, thermycin, and then known inhibitors. So these inhibitors can be used actually as drugs to inhibit the enzymes, and we have known inducers as well. Some interesting things to look here is that ethanol can actually induce certain CYPs. Same thing as caffeine can be substrates and also inducers, as well as tobacco smoke. So environmental factors are in play as well. So not only do you have to think about the drugs that these CYPs will work on, but also the environment that these CYPs are exposed to will alter their activity.
Will there be questions as to
Which with which? No. No. That's for somebody else to teach you, not me. You'll be getting that later. So these CYPs are all polymorphic. So polymorphic forms of some CYPs are found in the population, meaning that you have slight sequence variation in every individual. And so when we look at this, it can affect severely how people can respond to drugs. So some people might be more sensitive to a drug or more resistant to a drug because the form of the cytochrome P450 that they contain in their genome is either more or less active. So this can also account for drug-drug interactions. For example, warfarin, which is used to prevent blood clotting, is metabolized by the cytochrome P450 2C9. Phenobarbital is used to treat some forms of epilepsy, and it is a known inducer of CYP29C. So if you were to administer these together, the warfarin will usually be more metabolized quickly than you would expect because of the presence of that phenobarbital. So knowing what drugs your patients are on, you can find these drug-drug interactions, and some of which might be explained by the actual metabolism of the drugs themselves rather than the actual actions of the two different drugs. So let's have a little bit of a review question. Cytochrome P450s add what kind of functional group to a substrate? All right, we have a majority of class site hydroxyl group, and that is correct. Remember, these are monooxygenases, so they're going to be adding an OH group onto their substrate. So once we have this hydroxyl group on there, it's now going to go in through phase 2 metabolism. So these are all conjugation reactions where we're going to introduce highly polar conjugates onto our substrate molecules with the purpose of either detoxifying or make them available for excretion. So usually the site of attachment is often introduced by phase 1. So phase 1 typically will happen before phase 2. There are 5 specific types of phase 2 reactions which are important to remember. Glucuronidation and sulfation increase solubility. Then there's conjugation with glutathione, acetylation, and methylation. Now the last 3, the glutathione, acetylation, and methylation conjugation reactions, all help to detoxify and to inactivate. They don't really affect the solubility of the actual of their substrate xenobiotic molecules. It's only the first 2 which will really affect the solubility. So let's start off with glucuronidation. This is the most frequent conjugation reaction. UDP glucuronic acid, or abbreviated UDPGA, is added to the substrate. What it looks like is this. It's a sugar molecule, which is modified. And then it's attached directly onto the xenobiotic by an enzyme called UDP glucuronal transferases, or UGT. The attachment site is on hydroxyl groups, so of course you need that phase 1 to have that hydroxyl group put on. So a lot of their substrates include things like alcohols, phenols, enols, and hydroxyls and acids. And the oxygen site usually comes from that phase 1. Now, the UGT family is also very large since it's so common. There are about 19 known human genes. It has a wide distribution throughout the body, but they occur primarily in the liver. Of them, they're named very similar to the SIPs, where you have a family, a subfamily, and then the individual family member. So, example, UGT1A1 is family 1, subfamily A, individual member 1. And then there's many of these that are found in the liver, but there's also some that are very unique to the GI tract. And these GI tract are important in first-pass metabolism and will help to influence oral bioavailability as well. So UGTs are very common throughout not only humans, but throughout the animal world. One little interesting thing about UGTs are that in house cats, so UGT1A6 is the major detoxifying enzyme for drugs related to phenolic compounds that are found in plants. And so hypercarnivores, which don't eat plants at all, likely lost these genes during evolution due to their highly carnivorous diet. And so drugs that are related to these compounds are very toxic to them. So there are reasons in veterinary medicine why you'd never give acetaminophen or acetylsalicylic acid to cats or dogs or any other hypercarnivore, which are very toxic and, of course, would not be doing what you think it should be doing to the cat. And so, huh?
Is that your cat?
No, that's not my cat. That is like a demon cat or something.
What is it eating?
It looks like it's meat or something from the grocery store. Yeah.
It's a lot of sand.
So yeah.
It's a waste.
Anyway, so those are our UGTs. So the next one we'll talk about is a sulfate conjugation. This usually occurs on alcohols, arylamines, and phenols. So very similar. You need that OH group to add on the sulfate. These are catalyzed by sulfotransferases.
Sorry, 34.
Excuse me?
So I have 2 extra slides on here that you just skipped over. We don't need those.
No.
They're syndromes.
No.
Krigler and Najjar syndrome.
No, those we don't have time for those today.
Okay,
That's
then
fine.
Delete them. Then we're not
You can delete them. Yep. I didn't know they were still in there. That's all right. So sulfate conjugation occurs on the OH group, catalyzed by sulfotransferases found in the liver, kidney, and the intestine. So you have your drug, and then you're going to add basically a sulfate. The sulfate comes from what is called sulfate donor. So it's a molecule that the sulfotransferases will remove the sulfate and then attach it onto their substrate. The sulfate donor is adenosine 3-phosphate 5-phosphosulfate. No one ever remembers that name. Everyone remembers the abbreviation PAPS. So just use PAPS. This compound is typically also called activated sulfate. So this adding the sulfate group onto it will lead to an inactive water-soluble metabolite. And then glucuronate conjugation is a very competitive process. So there's a lot of compounds that will undergo this conjugation reaction. So the next one we'll talk about is the conjugation of glutathione to a substrate. We have seen glutathione before. This again is a small nucleophilic peptide, which is only comprised of glutamic acid, cysteine, and glycine. And again, that glutamic acid is not in a peptide bond with a cysteine. It is actually through its functional R group instead. And then this thiol group is what we're actually going to attach the glutathione to our substrate molecules during our reactions. So glutathione's typical job is to protect the cells and cellular macromolecules from interacting with electrophiles such as reactive oxygen species, reactive nitrogen species, and the thiol is what is actually going to bind to the electrophiles. So we use this glutathione with enzymes called glutathione S transferases, where we'll actually take the glutathione and put it right onto that xenobiotic. So the substrate are often epoxides and halogenides, and the conjugate is substrate for transporters for excretion. So these are specific transporters which recognize glutathione conjugates and then will pump it out of the cell. And again, these are the phase 3 reactions we'll talk about on on Friday. The next one is acetylation. So basically, you're taking the acetyl group off of acetyl-CoA and attaching it onto your xenobiotic, which will then give you just coenzyme A, which can go off and pick up another acetyl group. So the substrates here are usually primary amines and aromatic amines. The enzyme that does this is called acetyltransferases, where they will basically put the acetyl group from that acetyl-CoA onto their substrates. They're present usually in the cytosol of various tissues, particularly in the liver, and the acetylation does not increase the water solubility. So here's an example of a drug isoniazid, and then we have our acetyl-CoA, where then in the presence of the acetyltransferase, we then have the acetylated isoniazid here, and then we just have coenzyme A at the end. Our last conjugation reaction is methylation. This is an addition of a methyl group to a xenobiotic. The enzyme that does this is called the methyltransferase. It uses acetylcysteine methionine, or SAM, as the methyl donor. We have this molecule here is SAM. It's a rather large molecule. It's only going to take this one tiny methyl group and put it onto the substrate molecule. Here it's a catechol. And you can see the area in red is just where the methyl group was attached. So which of these phase 2 enzymes are paired with the correct functional group donor? Okay, so majority of class has sulfosyltransferase and with PAPs, and that's the correct answer. UGTs will always do UDP glucuronic acid as their donor, and then the other two options had the enzyme with the methyl donor, with the enzyme with their functional group donor switched. So methyltransferases use SAM, acetyltransferases use acetyl-CoA. So there are a lot of factors that will influence the actual rate and ability for people to metabolize xenobiotics. Some of which include age. So older people are less efficient at metabolism. They express less of these enzymes. And so that is something that has to be taken into consideration. The gender, sex, is linked to hormonal differences. There are functional differences due to the presence of estrogen or testosterone on some of these enzymes and due to gene expression profiles. And that leads to some xenobiotics being less metabolized in one gender over the other. And then there's also hereditary factors. So genetic differences can influence the amount and efficiency of the metabolic enzymes. We looked at this a little bit with the cytochrome P450s when we talked about the polymorphic nature, but many of the phase 2 enzymes also have polymorphisms, slight changes in their genetic sequence, which makes some people a little bit better, some people a little bit worse at metabolizing. And then there are also disease states. When you have liver disease, you have a lot of problems with xenobiotic metabolism because that's one of the major sites of both phase 1 and phase 2. But also cardiac disease and kidney disease can also have major effects on metabolism. So let's just look at one example of a drug and some of these genetic factors. So here we have a very common drug, codeine. And it's usually metabolized by CYP2D6 to make the active morphine. And then inactivated, when our body doesn't need it so much anymore, by glucuronidation to this compound called M3G, which is then excreted in the urine. Now, there are some people who have CYP2D6 polymorphisms, which makes it very much inactive. And so these people, they don't actually make that functional morphine, so they won't actually get the pain relief from the codeine. Instead, the codeine goes directly through glucuronidation to the M3G, which is inactive, and it's excreted. So this is one of the reasons why some people don't respond to codeine, is because their genetics just makes it so that they cannot actually get the benefits from taking that drug. Yes.
They'd still respond to morphine, though, right?
Oh yeah, they'd be fine with morphine. Yeah, or any morphine derivative not acted upon by that 2D6. But codeine itself specifically, it's just like nothing for them. So summarize the two different phases of metabolism. We have phase 1 with the purpose of enhancing the polarity, convert chemicals to a less toxic form, and convert chemicals to a more toxic active forms when there's that bioactivation. So there is that little caveat. It's not always a beneficial thing. And this is primarily cytochrome P450s. There are other phase 1 metabolic enzymes which we did not talk about. The major one is that cytochrome P450. Then phase 2 conjugation, we're going to make the xenobiotic molecules more water soluble. We're going to detoxify active groups. This is all due to conjugation type reactions, either glucuronidation, sulfation, conjugation with glutathione, acetylation, or methylation. So now that we are at this point, we have a molecule that is more water soluble. We need to get it out of the cell and into an area where it can be excreted, whether it's going to the feces or into the urine. And this is going to be done through specific transporters, which we'll talk about next time. Any questions before we go? All right, thank you.